Okay, so this is going to be another very high yield and important lesson for the IMAT exam. This part and the second part are going to be the explanation of the M phase of a somatic cell in your body. Somatic cells go through something called mitosis in order to duplicate the cell and the genetic material itself. We talked already about the cell cycle, and we talked about the fact that you have the S phase, which is the synthesis where you duplicate the genetic material. Then you have the G2 phase, when you have either the duplication of some things inside the cell that are going to be prepared for the mitotic phase. And then you have the M phase. Today we are going to talk about the M phase in somatic cells. The M phase stands for either mitotic or meiosis phase. This phase is crucial when it comes to the actual migration of genetic material to the different pores of the cell and the separation of the cells into two daughter cells. Mitosis is composed and comprised out of four main steps: prophase, metaphase, anaphase, and telophase. Each phase is composed out of many very important unique properties that you need to study and to understand. Prophase can be divided into only prophase. Metaphase can be divided into pro-metaphase and metaphase. Anaphase is only anaphase, and telophase is only telophase. After telophase, we have something called cytokinesis. Cytokinesis is where you have the separation of the cells. In animal cells, it will be called the cleavage furrow. In plant cells, you have the formation of a cell wall between them. Cleavage furrow only forms in animal cells. You don't have cleavage furrow in plant cells. We will study it in the future, but remember this very high yield fact for the IMAT exam from now, and the BMAT exams as well. Let's start from prophase. So during prophase, as you can see, we have an organism here which contains 4 chromosomes. So it also contains 2 chromatids for each chromosome, which means that we have a dipole creature. So the organism is 2N, and the dipole, the fact that it's a dipole, we abbreviate it as 2N. We have 2N equals 4, because we have 4 chromosomes for this dipole creature. Let's study this organism. So we have 1, 2, 3, 4 centromeres. Let's zoom in a bit. 1, 2, 3, 4 centromeres. Therefore, in this stage, we have 1, 2, 3, 4 chromosomes. We have 1, 2, 3, 4, 5, 6, 7, 8 chromatids. So we have 8 chromatids, 8 DNA molecules, but 4 chromosomes. In prophase, we have several things. The chromosomes actually become condensed, and you can see them as the karyotype. They become the most condensed way to actually to compact the genetic material. You have something called spindle fibers. Spindle fibers are microtubules that are coming out of the microtubule organization centers, the centromeres that we were talking about. So before the actual M phase, the centrosomes are going to duplicate, and you're going to have 2 centrosomes. So you're going to have the 2 centrosomes going to the separate sides of the cell. Don't forget that the centrosomes are comprised of 2 centrioles. So you have in total, during the prophase, you have 4 centrioles, 2 centrosomes. Each centrosome is in the other side of the other pole of the cell. I know it's a bit confusing, but bear with me. This terminology and this kind of this fact that it's a bit confusing, they really like to take advantage of it. So remember the following numbers, the terminology, and when it actually happens, where the duplication actually happens. Anyway, prophase, the centrosomes are going to be on the other pores of the cell, and from each centrosome you're going to see that microtubules are going to migrate towards the chromosomes, the dense chromosomes, and they are going to be aimed towards the centromeres of the centrosome, of the chromosomes, and you're going to see it in a minute. The nuclear envelope is going to start to break down, because in order to reach the genetic material, in order to reach the chromosomes, we need to fragment the nuclear envelope that is going to encapsulate and protect the genetic material. And the centrosomes that we just talked about are going to move to the other sides of the cell. Now, during pro-metaphase and metaphase, we have several main things that are going to occur. Chromosomes are continue to condense more and more. We have a protein called kinetochore that appears at the centromeres. And the kinetochore is the protein that is basically going to be the connection between the centromere and the macrotubule spindle. So it is going to allow the connection between them. It is, if we zoom in again, you're going to see that on top of the kinetochore, on top of the centromere, we have the kinetochores, which are as the following. Just a protein that is basically going to allow the spindle to be attached to the centromere. Also, of course, the mitotic spindle, which is the microtubules, is going to be attached finally to the kinetochore. So now you're going to see that this free spindle is going to finally be attached to the kinetochore. Now, the most important thing when it comes to metaphase is that the chromosomes are going to be lined up in something called the metaphase plate. And I want to wait a minute and analyze the following metaphase plate. So in this metaphase plate of the metaphase of mitosis, we have the 4 chromosomes lining up in the metaphase plate, because this creature, this organism is 2n equals 4. It has 4 chromosomes and it is a dipole. This is the most important part of metaphase. Then each sister chromatid is going to be attached to a spindle fiber from the opposite pole. So you are going to have this spindle attaching to one chromatid, and this spindle attaching to the other chromatid. You are going to have kinetochores attaching from each side, but you are still going to have a single centromere inside. So single centromere, 2 kinetochores, and 2 spindle fibers, and 2 chromatids, but 1 chromosome. Next, the anaphase. Now the chromosomes are finally going to split into 2. You are going to have sister chromatids, which are now called chromosomes, and you are going to see why in a second, pulling towards the different opposite poles. Then you have spindle fibers that begin to elongate in the cell. You will see it in a second. So anaphase, a very, very important thing. Now check it out what happens. Some microtubules are going to attach against each other to provide support to this process. Some microtubules that are attached to the kinetochore, which attach to the centromere, is going to dissociate the chromosomes that are now chromosomes, the sister chromatids that are now each one called a chromosome, to the other side of the cell. But why do we call it a chromosome now and not only a sister chromatid? Do you remember what is a chromosome by definition of this process? A chromosome is defined by the centromere. So now, because we separated the centromere and now we have 2 centromeres, one for each sister chromatid, each one is going to be called a chromosome. And now in this stage, after the separation, we are going to have 8 chromosomes and 8 chromatids. Unlike the metaphase when we have 4 chromosomes and 8 chromatids. Now we have 8 and 8. It's so important for the exam. It's really important. So remember, now we have 8 chromosomes because we have 8 centromeres, because we have 8 chromatids. So this is anaphase. Telophase is the process where the chromosomes are finally arrived to the opposite pole and begin to decondense. You have the nuclear envelope of each side of the new poles starting to form the new nuclear envelope. The mitotic spindle are starting to break down, and the spindle fibers are continue to push the poles apart to make sure they are not going to come together, just to provide the support. But now we have our next step. In order to provide the separation between the cells, I'm going to talk about specifically animal cells. Do you remember what we are going to do? We are going to form, using our microfilaments, our actin filaments, we are going to do something called a cleavage furrow. And a cleavage furrow is when you have the cleavage of the plasma membranes from each other, while you have the decondensing set of chromosomes on both sides. You're going to have the cleavage furrow starting to appear thanks to our microfilaments. So the microtubules actually separated the genetic material. The microfilaments are going to separate the plasma membrane. Now it's going to occur until we reach a point where we have two new daughter cells with the same set of chromosomes. I really hope this video made sense. I'm going to, in the next video, in the next part, we are going to talk specifically about the human cells and the human chromosomes. So I want to give you I want to make sure that you understand exactly what happens when it comes inside the animal cells when it comes to mitosis, because understanding what's going on inside the human cells when it comes to mitosis will really help you to understand the overall process of genetics. And then after a few videos, we are going to actually discuss and to learn about genetics.